Embryogenesis, gastrulation, neurulation, organ formation, larval development, and metamorphosis in freshwater fishes. Introduction. Embryogenesis in freshwater fishes is a highly regulated developmental process that begins with fertilization and culminates in the formation of a fully functional juvenile. This intricate process can be broadly divided into several key stages: cleavage, gastrulation, neurulation, organogenesis, larval development, and metamorphosis. Each stage involves precise cellular interactions, gene expressions, and morphogenetic movements that guide the formation of the fish body plan. Understanding these developmental mechanisms is crucial for aquaculture, developmental biology, and conservation biology. 1. Embryogenesis in freshwater fishes. Embryogenesis refers to the series of events following fertilization that lead to the development of a multicellular embryo. This includes cleavage, blastulation, gastrulation, and early tissue differentiation. Cleavage and blastulation. Cleavage is the rapid mitotic division of the zygote without growth, resulting in the formation of smaller cells known as blastomeres. Freshwater fish exhibit meriblastic discoidal cleavage, where cell division occurs only in the blastodisc at the animal pole due to the presence of yolk. Examples: zebrafish Danio and catla show rapid synchronous cleavage, leading to the formation of a blastoderm. Blastulation. The cleavage divisions result in the formation of a blastula, a hollow spherical structure consisting of an outer layer of blastoderm cells surrounding a fluid-filled blastocele. 2. Gastrulation. Gastrulation is the process by which the single-layered blastula reorganizes into a multi-layered structure with distinct germ layers: ectoderm, mesoderm, and endoderm. Cell movements in gastrulation. Invagination. The inward movement of cells to form the future gut. Epiboly. Expansion of blastoderm cells to enclose the yolk. Involution. Migration of cells at the blastoderm margin inward to form mesodermal and endodermal layers. Convergent extension. Cells elongate and intercalate to lengthen the embryonic axis. Delamination. Formation of distinct layers from an initially single-layered blastoderm. These processes collectively establish the body axis and define the basic body plan of the fish. 3. Neurulation. Neurulation is the process of neural tube formation, which gives rise to the central nervous system, CNS. Steps of neurulation. Neural plate formation. Ectodermal cells thicken and differentiate into the neural plate. Neural groove formation. The neural plate folds inward, creating a neural groove. Neural tube closure. The edges of the neural plate fuse dorsally to form a hollow neural tube, which later differentiates into the brain and spinal cord. Neural crest cells migration. These multipodal cells migrate and give rise to peripheral nervous system structures, pigment cells, and certain skeletal elements. Example: in zebrafish, neurulation occurs within the first 24 hours post-fertilization. 4. Organ formation, organogenesis. Organogenesis involves the differentiation of germ layers into specific tissues and organs. Development of major organ systems. Cardiovascular system. The heart forms from the mesoderm and begins beating within 24 to 30 hours in species like Danio. Digestive system. The endoderm gives rise to the gut tube, liver, and pancreas, which develop into functional digestive structures. Musculoskeletal system. The somites differentiate into axial muscles and vertebrae. Excretory system. Pronephric tubules form as the first functional kidneys in early larvae. 5. Larval development. After hatching, the larval stage begins, during which the fish undergoes significant morphological and physiological changes. Key features of larval development. Yolk absorption. The yolk sac provides nutrition until the larvae can feed independently. Fin formation. Median and paired fins develop progressively. Sensory organ development. Eyes, olfactory bulbs, and lateral line systems mature for environmental interaction. Example: in Labio rohita, larvae hatch within 24 hours and undergo rapid development. 6. Metamorphosis in freshwater fishes. Metamorphosis is the transition from larval to juvenile stage, involving structural and functional modifications to adapt to an independent lifestyle. Key transformations during metamorphosis: growth of scales, protective body covering develops. Fin differentiation. Larval fin folds transform into functional fins. Development of adult respiratory system. Transition from cutaneous respiration to fully developed gills. Changes in feeding habits. The mouth structure adapts to an adult diet. Example: in Clarias batrachus, larval to juvenile transformation occurs over weeks. Conclusion: the embryonic and post-embryonic development of freshwater fishes involves complex morphological and physiological changes governed by genetic and environmental factors. A thorough understanding of these processes has applications in fisheries science, aquaculture, and evolutionary biology.